Hello, and welcome to the RNA-seq-pop walkthrough. Now, if you found your way to this video, you probably already know what RNA-seq-pop is, or have some sort of an idea. But before I, jump into the walkthrough, I'm just going to briefly touch on what RNA-seq-pop is, and cover a couple of things that we can do with RNA-seq-pop. During the walkthrough, we'll talk about how to install RNA-seq-pop, what do we need to install, what input files we actually need to run the workflow. We'll go through the structure of the repository, the different folders. Talk about how to configure the workflow. So how do we choose actually which options of RNA-seq-pop we want to run, and then finally we'll set the workflow off running. So what is RNA-seq-pop? Well, RNA-seq-pop is a computational pipeline written in SnakeMake to analyze Illumina RNA sequencing data from any organism, and as well as performing standard transcriptomic analyses such as differential expression, gene set enrichment. RNA-seq-pop also can call and analyze genetic polymorphisms and extract some population genomic signals, like looking at genetic diversity or selection, for example. The GitHub repository is stored here. We'll take a look at that in a second, and then documentation here. The manuscript is currently as a preprint on bioRxiv. It's also being considered for publication at the minute. If you haven't read the manuscript, I would recommend that you do, first of all, as it should give some useful information on the kind of things that RNA-seq-pop can do. But quickly, just as an example, RNA-seq-pop can perform various quality control analyses. It can perform differential expression both at the gene level and the isoform level and visualize that data. We can calculate allele frequencies, mutations that we're interested in. As I mentioned, we can look at genetic diversity. We can also look at selection across the genome in the form of FST, or the population branch statistic. And if you're analysing enough of these Gambi, then RNA-seq-pop can do some extra things, such as looking at ancestry across the genome, so either Gambian Kalutzii or Gambi Kalutzii versus Arabiensis. And we can also look at chromosomal inversions and their frequencies in our samples as well, such as the 2LA and 2RB inversions. Awesome. Well, let's have a look at the GitHub repository. Okay, nice. So here is the GitHub repository. There's a link to the documentation just here, and the documentation website basically contains some of the same information that I'm going to be sort of talking about today, but potentially in a bit more detail. And if you've got any feedback on the documentation or on the workflow itself, please just let me know by raising an issue on GitHub or just emailing me directly. Cool. Well, the first thing that we want to do is we want to essentially install the workflow. But before we begin, we need essentially two things to be installed before we do this, but before we can run RNA-seq-pop at least. And those two things are Conda and SnakeMake. And so Conda is an open source package management system. It basically allows you to install lots of Python or non-Python programs into specific software environments. And so we need Conda because SnakeMake and RNA-seq-pop are actually going to use Conda to install all of the dependencies within RNA-seq-pop. So RNA-seq-pop uses a lot of different programs, and installing these ourselves would be just a massive pain, essentially. So we can get Conda to do this automatically, which is fantastic. But we need to have Conda installed. We of course also need SnakeMake to be installed, and the authors of SnakeMake encourage installation actually through Conda. So I would recommend that we first install Conda and then install SnakeMake. You might also find that when you first run the workflow, you actually need to install pandas. This is because essentially we load the sample metadata using pandas, and probably the easiest way to do that is with pip install pandas. Okay, so let's say that we've got Conda and SnakeMake installed, and now we want to actually get RNA-seq-pop onto our local system, or it could be our local system or a computing cluster. And so I'm going to copy, I'm going to go to here anyway, I'm going to copy this link, and we're going to clone that to our system. So let's open the terminal. I'm just going to put it in my home directory for now. So let's do a git clone. And we need to use recursive because RNA-seq-pop contains a git submodule, which is comp-careo. It's a program which is used to look at the different carrier types in and off with Gambi. And so we're going to use the recursive flag. Paste that link in. And we could do this directly, which would leave us with a folder called RNA-seq-pop. But I'm going to give it a different name. I'm going to call it RNA-seq-pop walkthrough, just so I know what it is. And it's quite useful to give the folders different names, particularly if you're going to use RNA-seq-pop on multiple data sets. You probably want to have different folders for each data set and give them a slightly different name. So let's do that. I'm going to call it RNA-seq-pop walkthrough. Unfortunately, the Git repository at the minute is quite large because it contains some large files in its history, basically, which are quite difficult to remove. So apologies for that if it takes a while to first install. It's currently about 100 megabytes or 120 megabytes. And so that should just take a couple of seconds more. Okay, so we should check. We should now have a folder called RNA-seq-pop walkthrough. And we can take a look at this. I'm just gonna, the first thing I wanted to talk about actually was, so actually, the first thing that we can do before we have any input files, before we have anything, if we want to, we can just check that it's RNA-seq-pop is set up correctly. And to do that, we already have a test data set within the repository. We can just run RNA-seq-pop on the test repository. I'm not going to actually fully run the data, run it. I'm just going to do a dry run. So you can do something in a dry run in SnakeMake, which is using the -n flag. And that will basically tell SnakeMake to figure out what it needs to do to run and what it will do when you run it. But it won't actually run the pipeline. So let's do that and see what happens. So you can see that RNA-seq-pop prints some information to the screen, such as where in your computer it's located, who the author is, that's me, what version of the workflow you're using, and the time that we executed this command, and also the dataset name. And we'll be able to provide our own dataset name for our dataset. Basically, this is just telling us it's going to install lots of different Conda environments for the different bits of analysis it needs to do. And it's going to run these rules. And so the rules in SnakeMake, you can imagine as each one is either like a command or a script, essentially. And it's going to tell us it's going to run all of these. There's a total of 96 commands or rules that it wants to run. These are the actual commands themselves. It's going to print lots of them. And here we have that again. And it also gives us actually the reasons why it's running the commands. So everything looks fine there. It looks like it should run okay. So that's good. So I wanted to talk about, first of all, before we look at the repository structure, is about what files we actually need for RNA-seq-pop. And they are basically a sample metadata file, which is in the form of a TSV, so it's tab separated. It needs to end in TSV. It doesn't need to end in TSV actually, but it needs to be tab separated. We need either single end or paired end RNA-seq data. So usually you will have paired end data, so you'll have two files, the forward read and the reverse read. And then we need our reference genome files for our organism that we're analyzing. And so we provide examples of the sample metadata file in the repository. So actually we can go into the config folder and there will be a file called example samples, and we're going to print that to screen. although it's not the most readable when you print it to screen like that. So let's have a look at this one instead. We essentially have 4 columns. We have the sample ID column, so each of our samples, you can see we have, in this experiment, we have 3 biological replicates. The treatment for each sample, so these are all the same treatment. Specifically, this is the control group from Tiacelae. Then we have its species, so these are Anopheles colucii, malaria mosquito. And we also have something called strain. Now, strain is not particularly important. If you don't have anything beyond species, please just fill out strain with the same information as species. And the reason we have strain is basically when we're performing principal components analysis, we do a principal components analysis for the whole data set, for each species, and then within each species, within each strain as well. So you need a file like this for your data. You also need, of course, your FASTQ reads, and essentially, there are 2 ways that you can specify the names of the reads. So either this can be done automatically, in which case they should basically be stored in the folder resources/reads, and they should be named as the sample ID _1 for the forward read .fastq.gz and _2 for the reverse read. And if you, in the configuration file, which we'll look at in a second, if you set the FASTQ option auto to true, SnakeMake will automatically look for these files in this folder. If you have like strangely named FASTQ files and you don't want to change them, it's a lot of effort, that's fine. What you can instead do is you can add 2 columns to this sample metadata file, one called FQ1 and another called FQ2. And these should contain the full paths to the FASTQ reads for each sample. When I say the full paths, it also needs to include, let's say if you put them in the resources reads folder, you need to include resources reads in the path as well. And to use that setting, you just change the FASTQ auto option to false. For single N reads, you just need an FQ1 column here. Okay. And also reads can also be trimmed with cut adapt if they need to be. So we have our sample metadata file filled out, tab separated. We also have our FASTQ reads either in the right place or specified specifically with FQ1 and FQ2 columns. We also need our reference genome files, and so we need a few things here. We're going to need a genome reference file that has the DNA sequence for each chromosome in FASTA format. And this can be gzipped. We need the same for the transcriptome in FASTA format again, and that can also be gzipped. We need a GFF3 format genome feature file, which just contains the information on exons and genes and their locations. And we also need one more thing, which is a sort of less common thing, which is essentially a file which maps genes to their transcript and also their gene name and gene description. And this is just useful for adding gene annotations to results and connecting transcripts to their parent genes. And so the GitHub repository contains an example one for Anopheles gambiae, and I'll also put example ones in for Anopheles funestus and Aedes aegypti as well. Otherwise, if you're analysing your own organism, you may need to make this file yourself. And then we also need a SNPF database name if we're performing variant calling. And then the last thing we'd need is not an input file specifically, but we need to provide a list of the chromosomes that we wish to analyse, or the contigs. And the reason for that is in some reference assemblies we have a few large or full chromosomes, but then we have hundreds of smaller contigs, and we don't necessarily want to analyse all these tiny contigs. So we sort of made it with an RNA-seq pop that you'd have to provide explicitly the contigs that you wish to analyse. Okay, so that is the input data that we need. Let's take a look at the folder structure of RNA-seq pop. So here we go. Okay. So you can see that we have 4 folders here. We have a license and a README. and the logo. So essentially, I'll go through these in order. So the config folder in here, it should have our configuration file, which should be named just not example config, but config.yaml. So basically, we need to edit this example and resave it as config.yaml. And we'll go through that in a second. And then we need our own samples TSV file. And again, it shouldn't be named example samples. It should just be named samples.tsv. We can take a look at that as well. You can see that a slightly different example to the one I just showed you, but the format is the same. And so we also have a docs folder, which basically just contains all of the essentially the documentation website is stored in there, so you don't really need to worry about docs. We have a resources folder, and this contains all the files that RNA-seq-pop is going to need to run, essentially. And so we already have some things in here. So for example, these two folders are ancestry informative markers for Anopheles gambiae, and a couple of other sort of annotation files for Anopheles gambiae. We have an example file that we can use to look at mutations that we're interested in. So if you're analyzing your own species and you want to look at some mutations, you just need to make it fit this format. And you can provide the path to that in the configuration file. And this is the example genes to transcripts file that I just spoke about. We also have the reads folder. Now currently this is empty. I am going to change that now. So fortunately, I have already got some reads which I want to use to hand. And they are in a folder called Sanjay RNA-seq data. Let's have a look what's in there. And we can see that there's a load of FASTQ files in there. So I am going to copy the FASTQ files. And we're going to move those into the RNA-seq walkthrough. Resources, reads. Okay, beautiful. They're all there. Very nice. I'm also going to move the I've already set up a config and a samples file for this. So I'm going to move them to the walkthrough and put them in the config folder. Perfect. And the last thing we have is a reference folder. So if we go to our reference folder, okay, it's empty. So we know we need a few files. Unfortunately, I have already put some of them in here. So what we have is we have a GFF3 file, genome feature file. We have a gzipped chromosome FASTA file and a gzip transcripts file. And then we also have a GAF file, which is used in gene set enrichment analysis. So let's copy these and put them in the walkthrough. Resources, reference. Nice, there we go. And now there's nothing left in here. We can quit this for now. Okay, so we've got these files here. That's cool. Awesome. So that's all set up now. Imagine that you've put your own files in here. Yeah, the next thing that we probably want to talk about is how to configure the workflow. And to do that, we need to open up our config YAML. So in your case, you will have an example config YAML in your repository, and you need to go through that and edit it to fit your own data, and then resave it as just config YAML. Now, I did this a few days ago. I've actually made some updates to the workflow recently, and so there's a few things that are actually wrong with this config file. Rather than redo this video, I'm just going to quickly, well, hopefully quickly change them. Yeah, I'm just going to quickly change them, and I'll see you in a second. Cool, so I've updated now. I've updated this config file to the latest version of RNA-seq-pop, and let's have a look at it. So there's information on every parameter in here is found on the documentation website. Let's take a look at that, in fact. So here in this on this page, we basically have each parameter. We just sort of describe each thing and what it does, essentially, hopefully in detail. If you have any questions with this or difficulties setting this up, please just let me know. So, here we are. So, configuring the workflow. So we need to edit this config file, and essentially there are lots of different parameters. RNA-seq does have a lot of different modules. Try to make it as simple as possible, but we essentially have a metadata file which we point to. So it's usually called samples.csv, located in the config. We give the dataset a name, and this can be anything that you want. It's just used to name a couple of output files. So just call it something appropriate. We're gonna use the automatic naming of the FASTQ files. So let's have a look. ls resources reads, and we can see that actually the way that these are done is it? Oh yeah, and the way that these are done, it is automatic naming. So we can see there's a sample ID underscore 1, 2, and so on and so forth. And this is indeed paired end data. So we put in the context we want. We're going to point to all of the reference files that I just talked about. So you can see that I can do ls resources reference, and we can see here that those files are present. So that's cool. I'm not going to go through all of this in complete detail. Essentially, we have options to run various quality control analyses. The contrasts is particularly important parameter, and this is when we're doing differential expression analysis or FST analysis, we need to compare two treatment groups. And so these are the different contrasts or comparisons that we'd like to do. So in this case, we'd like to compare. So if we also get up the samples file, let's move that to the side. We can see we have a few treatment groups: CONT-TIR, MALATIR, PIRI-TIR, and essentially here we're comparing the controlled TSLA group to the malathion TSLA group. And the way that this is done is that the case, or the in the case of insecticide, the susceptible group should be first. So the case is here, and then the control group is second. If you have a positive fold change of, let's say, 5, it means it's overexpressed in the second group compared to the first group. And the only important thing to note here is that the treatment names can't have underscores in them. So because we separate these with an underscore, we cannot have underscores basically in the treatment names. So we specify our contrasts. We say whether we want to activate certain bits of the quality control. So to do that, we just change trues to falses. I'm going to keep that true. Do we want to activate differential expression analysis? And then within that, there are various modules. So we can make Venn diagrams. We can even produce heat maps of specific gene lists if we want. We can look and see, are there genes? If we have 3 comparisons, we might want to see, or 2 comparisons, we might want to see, is there upregulation of certain genes in the first comparison and in the second comparison in the same direction? And that's what this module does. We can do gene set enrichment analysis, and we can also then do various bits of analysis with variant calling. And so we can activate that here. And there's a parameter called ploidy. So the ploidy here, ploidy should basically be, if your RNA-seq data is from a single individual and they are diploid, like mosquitoes or like humans, then the ploidy should be 2. But if you're using pooled samples, let's say you've pooled 5 mosquitoes together, essentially we have 5 mosquitoes that are diploid, so we have 10 alleles in total in this pool, and so we put the ploidy as 10. It's quite common in insecticide resistance research to people to pool 5 or 10 mosquitoes. The chunks parameter just is saying how we sort of break, when we're doing variant calling, to speed it up, we break it into different sections so it can be parallelized. And so we want to use a certain amount of chunks. By default, this is 9. I would recommend just keeping that as it is. Occasionally, you'll do a run, and one genome chunk will take a bit longer than others because it might have higher coverage or something like that by chance. That can happen, and so increasing the number of chunks should help that be a little bit faster potentially. Do we want to run PCAs and summary statistics and selection analyses? And within selection, do we want to run the population branch statistic, which requires 3 conditions, which are 2 closely related and an outgroup. And there's more details on configuring all of this stuff in the documentation. We have options to run the ancestry analysis, to run karyotyping, to looking at allele frequencies of mutations of interest, and also we can make heat maps of different gene families, and we can also actually even look to see in Anopheles gambiae only to see if any of the differentially expressed genes are lying underneath selective sweeps in the first phase of the Anopheles 1000 Genomes project. Cool. So let's say that we've configured this as we want. I think for me it was configured correctly. We now want to try and see if we can run the workflow. So something I didn't mention is that currently, if we look in the folder, we have these 4 folders. We're actually missing 2 folders at the minute. One of them is logs and one of them is results. And so logs, well, both of these 2 folders will appear, they will be created by RNA-seq-pop when we run the workflow. And so logs basically contains essentially a log of each rule or each command that we run, and they'll be put into specific folders and files. And the results contains all of our results. And within that, there will be various different folders, one for QC, for example, one for differential gene expression at the gene level, at the isoform level, one for variant calling and variant analysis. So that all looks good to me. I'm going to try and run the workflow now and see what happens. So if we go to the documentation, we go to running the workflow, it should give us a nice example command that we can use. So we're going to run this. So we're going to run SnakeMake with 4 cores. You may want to do more if you're running it on a server. And we're going to use this parameter use conda. This is telling us, please install everything through conda so that I don't have to install it all. If we don't use conda, you'll certainly get errors because SnakeMake, the RNA-seq-pop will run. It'll be trying to use certain programs which just aren't there. And believe me, there are so many programs that RNA-seq-pop uses, it's unfeasible to install them all ourselves. Okay, so the other thing that the command said was, and then we had the -n, it was a dry run. So we weren't actually running the pipeline. We were just seeing what will happen when we do. Awesome. Okay, so these are all the rules that are going to be run with our current set of parameters. Essentially, RNA-seq-pop reads this config file and says, okay, the user has said that they want to do differential expression analysis. Therefore, we must produce these files for the user. Essentially, that's what happens, and this is kind of the result that we see. And there's a lot of things in here to do with, variants of interest, allele frequencies, ancestry informative markers, some QC steps, checking the inputs are correct, looking at coverage, differential expression, and so on and so forth. So I'm going to run this now, and hopefully it won't give me any sort of errors. It will just start running. And we aren't going to wait until this is completed because it might take a little while. Depending on the size of your data set, typically the transcriptomic, the standard transcriptomic analyses are pretty fast, the differential gene expression and so on. And it's really the variant calling step that is generally the slowest. Once the SNPs have been called, then the rest of the analysis is quite fast in terms of looking at selection across the genome, what genetic diversity or so on. And usually RNA-seq will take a few days or a week. It kind of depends on the data set. But the initial transcriptomic things, at least, should be relatively rapid. And so what we can see now is that Conda is giving us a little sort of a warning. That's fine, we can ignore that. And it's telling us that it's creating these Conda environments. So it's creating one which is specifically for SAMTools FADEC. So that's for indexing FASTA files. And it says it's downloading that, it's done that, and now it's doing one for SNPAF, which is for annotating SNPs that we've called. So that's it. So RNA-seq is now running, and if I were to leave that, it should run to completion, and you will be left with a logs folder and a results folder. Inside the results folder will be your results, and I will put on the documentation website at some point a bit of a tutorial on what the results folder looks like and how to interpret things in the results folder. For now, I will leave it there. I hope this has been useful. Please give me any feedback you can. And yeah, good luck in using RNA-seq-pop. Just contact me if you have any problems. Awesome.